All right, next up, and the last speaker before the break is Tuulia Vallius, who holds a PhD from the University of Turku in Finland in the lab of Professor Seija Grenman? Yeah? Grenman. Okay. My Finnish accent. She also holds an MD from the University of Turku and trained as a resident in clinical oncology at Turku University Hospital. She is currently a postdoc in Peter Sorger's group at HMS. And is interested in the melanoma tumor microenvironment using cutting-edge spatial imaging to uncover new therapeutic insights.
Thank you so much. Thank you for the opportunity to have me today here and tell you about my postdoc work that I've been doing at Peter Sorger's and Santa Carlos group for the past few years. And really, I've been focusing on melanoma initiation and progression using human samples. And I'm not going to tell you about immunotherapy response prediction here, but I hope that during this talk you will get sort of sense of a framework that we could apply then analyzing these features in the immunotherapy treated patients in the future. So really, I'll be telling you a little bit background on my prior work in spatial profiling in melanoma, and then present you this cohort of samples that we are using for tumoral spatial profiling. Then let's go through 3 main points of the paper that I'm currently writing, and then summary and future directions. So this schematic here is representing our current understanding of melanoma initiation in humans when we don't have any underlying distal activity. So we start with atypical melanocytes that form these very invasive melanomas in situ. Then when tumor cells are able to penetrate the epidermal junction, they form radial and vertical growth phase melanomas. Vertical growth phase melanomas can actually then later on disseminate to lymph nodes and distant sites. And several melanocyte intrinsic and extrinsic changes are needed to transformation. First of all, of course, the acquisition of oncogenic driver mutations as well as other genetic alterations is very important, as well as epigenetic changes have been described to be a hallmark of melanoma initiation and progression. But then lastly, also very important feature is the escape from epidermal niche, escape from the location where you originally should be, ability to evade the immune response, and the tumor microenvironmental remodeling that is sort of forming a favorable environment for tumor growth. So lineage plasticity has been shown to play a major role in metastasis and disease progression in melanoma. And this is a very simplified cartoon. This has been very extensively studied using preclinical models and metastatic samples from patients. And basically, in a simplified way, what we know is that there's this proliferative state that is characterized by MITF-driven signaling, has high expression of pigmentation-related programs, and is still resembling the cell of origin melanocyte. And then through transcriptional reprogramming, these cells can undergo switch towards an invasive state that are characterized by these de-differentiated neuropressed-like embryogenic mesenchymal gene expression programs, loss of MITF-driven signaling, loss of pigmentation-related programs. And what can actually trigger this switch is exposure to therapeutics. So targeted therapy, immune checkpoint blockers can induce this switch towards this de-differentiated cell state, but as well as adaptation to the surrounding environment. So inflammation, hypoxia, oxidative stress can induce or trigger cell state switch. So in order to understand immunotherapy response and resistance in melanomas, actually, we need to understand the organizing principles of these tumors and need to understand how these features are present in untreated samples. So for this work, our aims were really to understand how early during melanoma development is de-differentiation emerging. Are different tumor cell states and intratumoral heterogeneity actually linked to specific tumor microenvironmental features? And could we identify a developmental trajectory associated with intratumoral spatial heterogeneity? So we started to do this work already a couple of years back, and this is our first cancer discovery paper describing this melanoma precancer atlas, where we deeply profiled one tumor using cyclic immunofluorescence imaging as well as microregional translocation profiling. And we really want to understand the arrangement, the spatial arrangement of these tumor cell states, as well as the microenvironmental changes over progression. And when we started to look at these tumor cell states in this one sample, we had a bunch of tumor intrinsic markers. Here I'm showing you S100B and MITF, and you can see how these expressions patterns are actually have this gradient type of expression. So we took all the tumor intrinsic markers, we applied spatial lag analysis, so this sort of like one type of clustering the tumor cells, and we received these tumor cell communities that you can then map back to their spatial locations. And when we look at one histologic region from this tumor sample, here I'm showing you invasive tumor front, you can actually see how spatially these different tumor cell states have this layered organization in this one particular region tissue. And then we want to understand that what is actually triggering the arrangement of these tumor cell states and what could be associated to it. So we measured here ERF1 staining in tumor cells, and we're showing here how sort of like there's this very layered pattern. These like tumor cells that are really close to immune cells at the invasive tumor front have the highest ERF1 staining. And here you can see with high resolution imaging as well, MI2 class 2 expression in these tumor cells, sort of like indicative of this interference signaling coming from potentially surrounding immune cells. So then we want to understand better the different features of invasive tumor front and what kind of immunosuppressive mechanisms are activated at a single time point in a single tumor sample. And we actually identified multiple different immunosuppressive mechanisms activated at the same time. So here I'm showing you one with high resolution imaging, PD-1, PD-L1 interaction, PD-L1 positive macrophages here reaching out to the 1 positive T cell. And you can see how these PD-1 and PD-L1 are co-localizing at the site of interaction. And really the length scale of this interaction is actually several cell diameters long. So really in order to understand the complexity and these different processes, you need different kind of imaging techniques that you can apply at the same time to these samples. So then in order to expand these observations from one sample to a cohort of samples, so with the help of Christine Lian, George Murphy, Eugene Semeno, we built this cohort consisting of primary melanoma samples. We had 62 samples, and here we are applying spatial transcriptomics as well as SISIF imaging for these samples to understand sort of the earliest evidence during melanoma development. And what we did for these tumor samples, we got these blocks. We sectioned these. So one section went for H&E and then other sections for SISIF. And then we did recap actually for microregional transcriptomic profiling geomics. And here you can see, so the H&E was used for pre-annotation of these samples. So we first confirmed the diagnosis. So here you can see the diagnostic feature for this patient was the vertical growth phase melanoma here in the middle. But actually, if you carefully evaluate these samples, you can see additional progression-related features adjacent to the diagnostic feature. So here, for instance, we have melanoma in situ, then we have strips of melanocytic atypia, and then at the end, there's like strip of normal skin. So then what we did with these annotations, we actually were able to map these back into the adjacent sections for SISIF, and we were able to analyze the SISIF data using these annotations, as well as we were using these annotations to guide the selection of microregional transcriptomics for sequencing. So in total, we got like more than 300 histological macroregions. And then for geomics, we collected sequencing data for 1,200 microregions across the cohort. So then we go to the SISIF data now first. We took the SISIF data, identified all the tumor cells from these images, and then we asked a simple question that what are the marker expression profiles for these each individual tumor cells and across the samples. And we use now here SOX10, MART1, SOX9, and NGFR, sort of like measure these different cell states and different combinations of these positive and negative markers. And what you can see here is that like once you hit towards like vertical growth phase melanoma, so that's the invasive component of the lesion. So there you see the loss of melanocytic tumor cell states and the gain of these neuropressile cell states. So really this dedifferentiation emerges in the regions of germline invasion. That sort of like makes sense. Here you see 3 examples from our cohort. So MEL14 here on the left, really high on MART1. One of the melanocytic markers. Then here in the middle, MEL71, half of the tumor cells have retained MART1, half of the tumor cells have lost MART1. You see this graded pattern in this sample. And then here on the right, you see sample MEL74 that actually has lost all MARC1 expression and is very high on NGF. And you can already pick here that there's a lot of heterogeneity in the staining of these different protein markers. And instead of these binary codes for positivity and negativity for these markers, you can also see gradients in the expression pattern for several of these markers, highlighting again the intra-tumoral heterogeneity, both in macro regions, but as well as in this micro region at single cell level. So then we took the geomics data that we had just collected and annotated, and we performed a very simple principal component analysis. And here we're plotting PC1, PC2, and each point here is now a single microregion from different patient samples. We're coloring these microregions based on the histopathological annotation, and you can see this clear trajectory, this progression trajectory from normal precursor regions all the way to invasion, forming nicely with the slingshot analysis. This part of analysis is done by TK Shi, who is a grad student in the lab here also present. And then we take the same data, same PC1, PC2, but instead of coloring by histopathological annotation, we took these previously published gene signatures and looked at the enrichment scores for these gene signatures across the data. And again, you can see here that these gene signatures linked to normal pigmentation MITF programs are sort of like forming this arc towards the top of PC2. And then there's a drop in the expression towards the tail in PC1. And then you see that there in the tail, that is really low on MITF pigmentation-driven signatures. You actually see a gain of dedifferentiation neural crest-like signatures. And then here again, these three samples that we saw in the previous slide. So you see MEL14 here towards the left in PC1, more towards like normal. And then you see MEL71 mapping towards the top of PC2 and towards like right-hand side in PC1. And then at the tail, there's this NGFR high sample, MEL74, sort of like showing that these like staining patterns also like reflect what we see transcriptionally. So then now we can focus only on the first sample set that is like composing of only these like very early stage melanoma. So we remove the tail of the PC2 and like remove all the later stage samples and do the same exercise. We do principal component analysis, map PC1, PC2. And what we see here is again, there is this progression trajectory coming from normal precursor all the way to invasion. However, in the invasion, there you see a lot of variance between these microregions. Then when you start looking at individual samples, so the variance between the samples can be equally high as within each individual samples. And these samples are not only clustering based on the patient ID, but actually these are like more following these spatial patterns where these microregions were coming from. And that also like reflects the staining patterns that we see in these microregions. So sort of like it's notable that this variance within this transcriptomic data can be equally large intratumorally as well as intratumorally. So then now that we established that there is a lot of intratumoral heterogeneity, so how could we actually quantify, measure this? So Shannon entropy would be a classical way to measure diversity within a biological system. So it takes into account the amount and abundance of different cell types. However, since tumors and tissues in general are very organized structures, so we need to have the spatial component included. So what a former postdoc in our lab, Edward, did was that he extended the Shannon entropy. So he included amount, abundance, intermixing, and distances of various cell states. So what that gives you as an output is single score, like single value for a microregion or macroregion, the region that you are interested in studying. And here you can see a subset of our samples being scored for spatial entropy. And again, you can see the high variance between the samples. Here's one example of a sample that was scored to have like high spatial entropy. And you can see these different cell states intermixed in that sample. So you can do the same exercise for these microregions. And again, you see that in each individual patient, again, there's a lot of variance between the entropy scores when you calculate the entropy scores for these microregions, for the geometric regions. And then when you try to map entropy to progression axis, so what we saw was that like when we calculated spatial entropy for regions of melanoma in situ and regions of invasion, we see that on average, the mean entropy decreases over progression. However, when you calculate entropy scores for these dermal invasive regions and you bin these samples based on Breslow depth, you actually don't see an increase in mean entropy along like Breslow depth, but what you do see is the increase of variance again. So there can be like very deep invading tumors that can be very heterogeneous and very invading tumors that can be very, very homogeneous. So then we decided to take like one step back and actually think of more the de-differentiation and sort of like the spatial component. And we took this MART1 loss as surrogate for de-differentiation in melanoma. So we took all the single cells from our SciSIP data, and then we determined the spatial neighborhood for each individual index cell. Here we're using like a radius of 15 micro. So it's really like the small neighborhood, immediate neighbors surrounding you. And then you select the data to include only tumor cells, and you define these tumor cells into MART1 positive and MART1 negative tumor cells. And then you ask the simple question that how are these neighborhoods different between these two groups? So what you do see is that these MART1 negative cells are actually more frequently surrounded by these immune and different stromal cell types compared to tumor cells that have still retained MART1, sort of like suggesting that the loss of MART1 is really occurring in these highly organized like spatial niches. So as a summary, what I showed you now is that tumor state changes follow this histopathological progression axis with high intra and intratumoral heterogeneity. Spatial intratumoral heterogeneity can be measured with entropy, and de-differentiation is a spatial feature in melanoma. So then what to do in future? So we have other avenues as well that we want to explore, but in the context of immunotherapy response and resistance, so we could start like measuring these different features from baseline tumor samples, of course. But then the challenges are still like several, like we really don't know like which features are the main drivers of outcome. Which features should we like focusing on? Is it like single feature, or is it like the complexity, the totality of different features in together? So this is something that we have to figure out. Even simple measurements, such as like quantification and classification of PRISC TILs and non-PRISC TILs, is not very established in melanoma. So we still have a lot of work to do. Of course, access to patient samples within clinical trials is very critical point that would need to be sort of like overcome. Spatial data collection, especially with like these new techniques such as Xenium, Visium type of approaches, is very expensive. SciSIP is very cheap, but still like spatial data analysis is actually very labor intensive. So that brings the expense. And it really requires collaboration of dedicated pathologists and biologists to really understand what is going on there within these tumors. So with that, I'd like to thank everyone who has been involved in this work. It's been a great team effort. And the funding organizations and of course, patients and their families and caregivers for providing these samples to address these difficult questions. And I'm happy to take any questions.
Yeah, I did have a question. I kind of lost in the last point about the cell heterogeneity and the MIF1. It seemed like MIF1 minus cells just generally had more cells around them versus MIF1 positive.
Yeah, so this
or MART1,
yeah,
excuse me.
No, good question. So really, what I'm trying to show here is that these MART1 negative tumor cells were locating in these immune high niches and stromal high niches. So compared to the tumor cells that had retained MART1 that were more frequently forming the cold tumor. But what I didn't mention you is that like then you could ask that, okay, where there's really like at the invasive like tumor edge, like the leading edge, and that's not what we saw. We saw that it was like immune association, but when we try to measure by depth or when we try to like define these like invasive tumor fronts, it's not actually the case.
And I had a second question. Is there like a time component in your single cell trajectory against time a factor? Is there like a convergent evolution? It always kind of goes to this neural crest phenotype as a function of time?
So I would say that we haven't looked enough samples, right? So we collected 62 samples now. It's a good representation of melanoma, but I would say that we still need to look a little bit more to understand sort of like where are the branching points, because like the literature is like pointing out that like in addition to neural crest like phenotypes, you also see these axial high, these like really undifferentiated phenotypes like mesenchymal phenotypes. So I think like we're now a little bit biased by the data that we have, right?
Can you tell us a little bit more about the data? So how large are these tumors?
So let me show you one example here. For instance, here. So this is like stage 1 melanoma. So you can see the scale bar here, 500 microns. So these are really small samples. And that is also like in a way one caveat. So the cell numbers in these early stage melanomas are very small. And then the other caveat is that like no one would ever do single cell sequencing upfront for these samples because these are really diagnostic specimens. So small tumors, and we are limited with these like FFPE approaches right now.
And how can you get access to that, given that they might still be needed for diagnostic visits later on? Is that not an issue?
Yeah, so this is like sort of like considered to be like clinically like discarded material at the moment, like what we are working for.
Oh, because they're old, but because
These,
they're small.
well, these are like they always like reserve like certain subsets of like tissue for like additional diagnostic like purposes if needed, but like sort of like we are being given like leftover material.
Yeah.
So it's really interesting that you're picking up these MART1 positive, or at least the MART1 negative MITF low cells having more immune infiltration. And because I think there's a dogma, as you're aware, that these undifferentiated melanomas might be more resistant to kind of conventional therapies, including targeted therapies, immunotherapies. So do you have any sense about the effect of immunotherapy on each of these populations? For example, in paired samples, do you see enrichment or depletion of these undifferentiated cells?
So that's really, really good question. And I think that still requires more data to understand it better. What we've done in the context of in-transit melanoma, where we had longitudinal biopsies from single individual patients who maybe underwent like immunotherapy. For instance, like there in one patient, we see that from earlier, there might be like subset of these differentiated cell states present, but then like we really see these like highly like differentiated tumors like during therapy. So in a way, I think I don't have a clear understanding. What I do see is that like these primaries are already very complex, even without any like therapeutic intervention, right? So, but I think that's a good point that like how often do we see that in therapy, for instance, like we have this like bottleneck, this purification of specific cell states, right?
Yeah, I think you could see, like, given that your macrophage population, for example, is higher in the MART1 negative, you could argue that they are actually going to be resistant despite this immunization. But then there are more CD8 cells in the same population.
Absolutely. I think like what the data is sort of like pointing out is that like targeting these individual features is not really the effective like way, because like there are multiple other features that sort of like also might play a role, right? So in a way, how do we design sort of like therapeutics that are able to hit like multiple different targets at the same time, right?
Different question. Just there's methods being proposed now that try to extract gene expression and other complex features from H&Es directly. And I guess I'm wondering if you think that's a plausible way of getting additional data, because those are so routine, or if it's possible to sort of train like a translation. You use this high dimensional data to find the biological mechanisms, and then you train a kind of translation model to then extract surrogates of that from digital H&E slides.
So I'm sure that there's like sort of like a lot of potential in those approaches, and in a way like again like we have to like maybe carefully again like explore these in order to understand like how like usable they are in the future. But like in a way there are still like many fundamental things that we don't understand from H&Es. For instance, like how does morphology like reflect these like different like cell states, like molecular cell states that we are able to like see with imaging, right? Or like how do H&Es sort of like reflect of the different immune types that we can sort of like detect by like immunofluorescence imaging, for instance. So in a way, I think that that's like sort of like a great like future goal, but like we need to sort of like understand these simple questions first in order to understand the very complex question, right?
But I think in answer to this question, there's ongoing work in Sober Lab to elements of this for more rudimentary features. So for instance, identifying mitotically active cells and then training the HME to detect them better, because the HME really overlooks it. We can easily identify only one part of mitosis visually by an eye, whereas if we apply some of these markers, we can actually go through the whole page.
Gotcha.
And then be able to identify it more sensitively.
You
So-
can get extremes.
Correct, exactly right. So for individuals, easy features, you can already do this, but to Tuulia's point, if you're looking for more complex features, it's going to take a lot more work, and some of them you may not fully be able to train.